it may be kind of odd, but ask any immunologist or microbiologist or virologist about their favorite immune cell, and they'll probably give you an answer. Though I've gained appreciation over the years for the role of T helper cells and for cytotoxic T lymphocytes and for the role of NK cells, my heart is still with the role of B cells in producing antibodies in what we call the humoral immune response. In this lecture, what we're going to talk about is how B cells become activated, how they function to produce antibodies, and then we'll focus specifically on some of the roles of antibodies in controlling an infection. Now, in a previous lecture, we talked about how T cells are made in the bone marrow and then selected within the thymus, which is where the T comes from in the name T cell. B cells also undergo a development and selection process, but all of this happens for B cells within the bone marrow. And similarly to what we see for T cells, the B cell receptor is going to be selected or tested in a way to ensure that it will respond to foreign or non-self epitopes, but not to self epitopes. The benefit of this is it greatly reduces the likelihood of autoimmunity and also reduces the likelihood that B cells are circulating throughout the lymph system with defective B cell receptors, or what we abbreviate as BCRs. Now, one of the really cool things about B cells is not only do they secrete antibodies, but they are professional antigen-presenting cells. And this puts them in the same class as macrophages and dendritic cells. And so what B cells are going to do is that they are going to present antigen on MHC2, and they're going to present it to T helper cells. And in fact, that's how B cells are going to communicate with T helper cells. What's different, though, is that B cells bind to antigens not through molecular pattern interactions with PRRs, but rather through a specific interaction between a B cell receptor, which is shown on the right here, and a specific epitope. You may note, looking at this picture, that the B cell receptor has some similarities to the T cell receptor. Like the T cell receptor, there are constant regions that are going to be the same in all B cells. There is a variable region, which is going to be unique to each B cell receptor that is made. And in fact, the generation of this variable region is going to be the same type of generation that we saw with T cells. In other words, the process of genetic rearrangement and hypermutation of what we call the VDJ region of the genome is going to lead to the generation of this unique B cell receptor. Now, these variable regions then contain the antigen binding sites. And what you may notice here is that whereas in the T cell receptor there was only one antigen binding site, well, B cell receptors have two of them. Keep this structure in mind because we're going to come back and talk about antibodies in a little bit. And the antibody is essentially a non-membrane bound B cell receptor. Now, let's take a look at how B cells are activated. We're going to assume that the cell on the left-hand side here is a T helper cell that has already been exposed to an antigen and it has been activated. The cell on the right is a B cell that has not yet been activated. Now, these B cells have their B cell receptor, which I've shown in blue here, and again, these bind to specific epitopes. And so this particular cell, as functioning like a professional antigen-presenting cell, will bring that pathogen or that antigen into the cell, perform phagocytosis, and then present that antigen or epitopes of it on MHC2. Now, if this T helper cell recognizes that epitope, it responds by secreting cytokines. And these cytokines are going to stimulate and activate those B cells. Now, one of the things that B cells do in response to this cytokine stimulation is that they differentiate into what we call plasma cells. In this image, I'm showing a plasma cell, and I'm trying to highlight here the ER in orange and the Golgi apparatus in blue, because plasma cells really change their gene expression in a way to mass produce antibodies. They secrete these antibodies, and so they have to have a lot of the organelles that contribute to the endomembrane system. The other type of B cells, well, they're memory B cells, and these memory B cells are going to remain rather quiet until the next time they see a pathogen that they recognize. And so this B cell receptor here still would have that membrane-bound B cell receptor on its surface. Now, here is the structure of an antibody, and the type that we're going to talk about in particular is known as IgG, which stands for immunoglobulin G. In total, there are 5 different immunoglobulins that our B cells will produce, and the membrane-bound B cell receptor is one of those forms. It is known as IgD. And so, in fact, the B cell receptor is a membrane-bound antibody. And when B cells differentiate into plasma cells, they change the type of immunoglobulin or antibody that they produce. Now, that antibody maintains its variable regions, but what changes is this constant region. And this constant region changes from one that's membrane-bound to one that now can be secreted from the cell. Now, as we already noted, all antibodies are going to have 2 antigen binding sites, and you can see in the cartoon on the left-hand side and also in the image on the right, these antigen binding sites contain 2 chains, a light and a heavy chain. But what's more important here is that these arms that make up the Y of the antibody, they're flexible. They can move around. And so this allows for antibodies to bind to the same epitope twice. Now, another term that I'd like you to at least be familiar with, because this is going to become important down the road, is what's known as the Fc region. This is also sometimes referred to as the stem of an antibody, and this is very important because it's going to help facilitate the innate immune response to pathogens. Now, antibodies perform a variety of functions that we usually group into one of 5 categories. And we're going to review each of these. 4 of them are ones that I would expect you to be more familiar with or probably feel that they make sense. The last one we're really not going to focus on because it's not something that we've talked about with this unit. The other thing to point out is that these activities or these functions of antibodies are not exclusive of one another. Oftentimes, we use these names to describe specific features of them, but to be honest, they are overlapping functions. So let's look at the first 2. On the left-hand side here, I'm showing you an antibody that has been bound to a virus, particularly the spikes, to the flagellum of a bacterium, or to a toxin. And these all represent forms of neutralization. In neutralization, an antibody will prevent the antigen from attaching to a host. We know that the viral spikes are required for viruses to attach to host receptors. The flagellum on bacterium is also an attachment factor. And the B portion of this diphtheria toxin is required for the diphtheria toxin to attach to host cells. The next mechanism is known as agglutination. And agglutination basically means to clump. Well, what's happening here, as you can see in this case here between the bacterium and the IgM antibody, the bacterium are being clumped together. This is going to have a couple of effects. One, it makes phagocytosis easier. This process will make it easier for phagocytes to engulf the pathogen. Secondly, it will help the spleen and kidney to filter the pathogen from the blood. The third mechanism is known as opsonization. When a pathogen is opsonized, it is basically coated with antibodies. You may say that this sounds a lot like neutralization, but the goal here is to increase the binding of the pathogen, or the antigen, to phagocytes. You may recall that phagocytes have pattern recognition receptors, or PRRs, that they use to bind to pathogen-associated molecular patterns. Well, phagocytes also have another type of receptor on their cell surface known as the Fc receptor. And I've already mentioned that the Fc region of an antibody is the stem region. What the Fc receptor, or the FcR, does is it binds to this Fc region of an antibody. In this indirect way then, as you can see in this picture here, the macrophage, by binding to the antibody, which is bound to a pathogen, will be able to phagocytize this pathogen and destroy it. An antibody-dependent cellular cytotoxicity, or what's also known as ADCC, antibodies will bind to a pathogen and they will stimulate NK cells or eosinophils to secrete granzymes and perforin. You can see in the image below that this image is showing that NK cells have an Fc receptor, just like we saw with macrophages in the past slide. When this Fc receptor binds to the antibody that's bound to the antigen, this stimulates the NK cell to secrete the granzymes and perforin that would be used to cause cell death. Eosinophils perform a very similar function, but it is thought primarily that eosinophils are used to treat parasitic worm infections such as tapeworm and heartworm. Now, you know that I would not be doing my job unless I talked a little bit about how pathogens can avoid this humoral immune response, and Staph aureus is a great example. You may recall Staphylococcus aureus is a gram-positive bacterium. It forms into these staphylo clusters, and then the coccus describes its spherical shape. And this particular microorganism produces a protein that is known as protein A. Well, one of the really interesting things about protein A is it binds to the Fc region of an antibody. And we just talked about how important this Fc region is for opsonization and for ADCC. And so if antibodies are secreted, what can happen is that these proteins, protein A here, will bind to the Fc region of the antibody, and this has two effects. First, it prevents the ability of the antigen binding site, those arms of the antibody, to bind to Staph aureus. Secondly, it prevents phagocytosis and opsonization of this microorganism, therefore allowing it to avoid our immune detection systems. All right, so in summary, let me kind of cover here three major points that we've talked about in this particular lecture. The first is that B cells are professional antigen presenting cells. They're produced in the bone marrow, they undergo maturation in the bone marrow, but they are activated by T helper cells and their interaction with those T helper cells in lymph nodes. B cells produce antibodies, and they produce large quantities of these antibodies. And that differentiated B cell that produces an antibody is known as a plasma cell. B cells can also differentiate into memory cells that will help against subsequent infections from the same pathogen. Finally, we talked about the roles of antibodies, and we focused on 4 major roles here, that being their role in neutralization, agglutination, opsonization, and antibody-dependent cellular cytotoxicity. Hope to hear from all of you soon. Take care.